Welcome to tutorial 23. Our topic today is proteins. Before we get started, just a quick reminder that the notes to accompany this video tutorial are available online through my website. And with that said, let's go ahead and get started. So proteins perform a variety of different functions within a living organism. And I've written up several of those functions on the board. These are also written for you on slide 2. We're going to talk about each of these roles just briefly right now. So almost all enzymes are proteins, specifically globular proteins. So let's write that up here. Almost all enzymes are globular proteins. And if you don't know what an enzyme is, don't worry, I'll give the short and simple definition right now, and then in tutorial 24 we will actually dedicate our whole topic to enzymes. So an enzyme is a biological catalyst. What does that mean? That means that enzymes are the molecules that our body relies upon to speed up the rate of all those different biochemical reactions that keep us alive. So a very important class of molecules, to say the least. All right, hormones. Many hormones are proteins or smaller versions of proteins called peptides. So let's write that up here. Many are proteins or peptides. Our structural tissue is largely composed of proteins. So our hair is made up of proteins, our nails, our skin, our muscles are largely protein. Collagen, which is responsible for our connective tissue, is a protein. So structural tissue here, I'll just write some examples: hair, nails, skin, muscles. And transportation of many molecules within the body is due to the use of proteins. We saw this back in tutorial 22 when we talked about facilitated diffusion and active transport, two transportation methods that require the use of embedded proteins within cell membranes. So let's write down here, for example, examples: facilitated diffusion and active transport. Both utilize proteins within cell membranes. We also briefly talked about the use of lipoproteins to transport cholesterol through the bloodstream. So again, another example where proteins come into play in transportation of a molecule. So let's write that down here as well. Lipoproteins. Okay. And immune response. Last but not least, immune response. Our immune response is the result of the use of specialized proteins called antibodies, and their job is to identify and attack foreign invaders within the body, like bacteria and viruses. So let's write down here that antibodies are proteins. Okay. So it really is amazing that this one class of biomolecules is responsible for such a wide variety of different functions in the body. So what is it that makes these proteins so incredibly diverse in their function? Well, it really all goes back to their structure. The structure of the protein dictates its function. And so that's what we're going to be focusing on today, the structure of proteins. So let's erase here and go on to slide number 3. Proteins are made up of smaller individual units called amino acids. And these amino acids get linked together via amide bonds to make up the larger peptides or proteins. Now there are just 20 different amino acids that make up all the different proteins in living organisms. Just 20. And we're going to talk about those amino acids first. Okay. All amino acids have the same general structure. They have an amine group, kind of as the name implies, right? Attached to a carbon with a hydrogen, and then something else that I'm just going to leave blank for now, because that's the part that can vary, and a carboxylic acid. Okay. Then for this fourth bond to this alpha carbon, we're going to call it the alpha carbon because it's the carbon next to the carboxylic acid. That's the part of the amino acid that can vary. And I'll just write R here, because this R group can be a variety of different things, and that's what makes up the 20 different amino acids. So let's write here that this R group can vary. Okay, so real quick, just kind of a side note, notice that we have an acid and a base within this same molecule. So this acid and base can undergo an intramolecular acid-base reaction where the acidic hydrogen gets transferred over here. And so in some textbooks, you will see amino acids written in that ionized form, which is called the Zwitterion form. So let me go ahead and draw that here. So if we have that H plus transfer, that acidic hydrogen transfer, and we're going to end up with a negative charge here for the loss of the H plus, and a positive charge here for the gain of the H plus to that basic functional group. And again, this is called the Zwitterion form. So I'm going to label that over here. And the reason that I want to show that is because a lot of introductory textbooks are going to be drawing their amino acids in this Zwitterion form. I'll stick with this form, but don't let that throw you off if your textbook or your instructor is using this form. It's the Zwitterion form. All right, so we said that R group can vary. Well, what can it be? Well, we can categorize that R group into 3 main categories. That R group can be, let's do this in blue, that R group can be a neutral side chain, meaning that it's not charged. Okay, so some examples would be glycine, which has an R group that is just a hydrogen. So you can imagine just a hydrogen here. That would be the amino acid glycine. And amino acids will be abbreviated with a 3-letter abbreviation. So I'm going to write GLY for the abbreviation of glycine. That's a side group for glycine. Alanine is a neutral side chain, has a neutral side chain. Alanine side chain is a CH3. Abbreviation of alanine is ALA. And let's do one more. Serine has a neutral side group as well, which is just a CH2 with an O H, no charge. Okay, so serine, S-E-R for serine. Okay. Now within the neutral side groups, you can have two subcategories. Okay, so those neutral side groups can either be nonpolar, which makes them hydrophobic, water-fearing, or they can be polar, which makes them hydrophilic, water-loving. They will interact with water molecules. Okay, so we've got, and let's go to a different color for this. I'll go to the pink, because my red pen died. All right, so we've got the side group of glycine and alanine. We can see that those are going to contain just nonpolar bonds, meaning that they're going to be hydrophobic. Okay, so glycine is going to have just a carbon-hydrogen bond here. That's a nonpolar bond. Alanine is going to have a carbon-carbon and then 3 carbon-hydrogen bonds, all nonpolar bonds. So hydrophobic side groups, neutral, nonpolar, and hydrophobic side groups. And then serine, we can see with the OH here that this is going to be a very polar side group. So this is a good example of a neutral but polar side group. So it's right here, polar, which makes it hydrophilic, water-loving. You can probably even imagine already this OH hydrogen bonding to water molecules. So very water-loving portion of the molecule. All right, so we said there's 3 categories. We just talked about one so far. The side group can be neutral, and those neutral side groups can be either polar or nonpolar. The side group can also be acidic. There are some amino acids that have acidic side groups. So let's talk about that next. Acidic. And a good example, there aren't very many, I think there's only 2, glutamic acid and aspartic acid. So let's use as our example aspartic acid because it's got a shorter chain. It's got a CH2 and then a carboxylic acid. And at physiological pH, the carboxylic acid of the side chain will be deprotonated. So I'm going to go ahead and indicate it with the 1- charge because it's lost as H+. Okay. So this is aspartic acid, called aspartate when it is negatively charged like that. And that is an acidic side group. And then we also have basic side groups. And again, not very many of these. Lysine is a nice simple example. It's got a CH2, 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 and then an amine at the end. And again, that basic side group is going to be protonated at physiological pH. So I'm going to go ahead and draw it as an NH3 with a 1+ charge because it gained the H+. It will have a 1+ charge. So that is the side group lysine, or the amino acid lysine. All right, so let's erase here. So when looking at that alpha carbon of our generic amino acid here, notice that that alpha carbon has the potential to be chiral. The only amino acid that is not chiral is glycine. And the reason it's not chiral is because the R group of glycine is a hydrogen. Okay, so then you'd have 2 hydrogens and you wouldn't have the 4 different constituents needed for a chiral carbon. All right, so let's write down here. All amino acids are chiral except glycine. Now we just said that's because glycine has an R group that is just a hydrogen. Okay. So if we look at our amino acids in that Fisher projection, That's the form that we learned when we were studying carbohydrates several tutorials ago. We can more readily see this chirality. So let's go ahead and indicate our one chiral carbon. Remember, that's indicated at the intersection of the two lines. We just have one chiral carbon, so I'm just going to indicate one. We put our most highly oxidized carbon at the top. That would be the carboxylic acid. We're going to put the R group at the bottom. And then the hydrogen and the amine can be either on the right or the left. So let's indicate this isomer as having the hydrogen on the right and the amine on the left. Well, then the opposite isomer, the enantiomer of this molecule, would then have the amine on the right, the mirror image, remember, and the hydrogen on the left. Okay. And remember that these molecules are not the same. They are mirror images that are non-superimposable. And remember that the vertical lines are going away from you into the plane of the whiteboard, and the horizontal lines are coming out at you because this carbon has a tetrahedral geometry. So there's no way that you can superimpose these two molecules. They are not identical. They are related as enantiomers, non-superimposable mirror image forms. Well, when we have, let's go to the pink pen, make it really stand out. When we have the amine group on the left in a Fisher projection, form, it is the L or the LAVO isomer. So this is the L isomer of the amino acid. When we have the amine group on the right in this configuration, in this Fisher projection configuration, it is the dextro or the D isomer of that amino acid. All right, and interestingly, nature selectively uses L isomers of amino acids to synthesize all proteins. So very interesting. Let me write that point down. Nature selectively uses L isomers of amino acids to synthesize proteins. Okay. All right, so I think we're ready now to go on and talk about how those individual units, how those amino acids are linked together to form the larger peptides and proteins. So let's erase here. Carboxylic acids react with amine functional groups to form amide bonds. And this is exactly the reaction that's going to happen between amino acids to form the larger molecules known as peptides and proteins. So let's write that up here. Carboxylic acids react with amines to form amides. Oops. Okay, so let's look at a generic reaction first between some generic carboxylic acid. That's why I'll just write an R over here, because it doesn't matter if it's acetic acid or whatever carboxylic acid it is. And it's going to be reacting with a generic amine. So I'll just write R2, and there's a hydrogen here. And I'm going to go ahead and put the lone pairs. And these reactions in the lab will require the addition of heat. So I'm going to write heat above the arrow, but you might recall from previous tutorials, I always like to remind you that in the body, these reactions are catalyzed by enzymes. So I won't typically write heat in the future because we'll be talking about biological reactions which are catalyzed by enzymes. Okay, and hopefully helpful to you, I'm going to show some arrows, but notice that these arrows are not representing a mechanism. These arrows are just here to help you remember where new bonds are forming and where some bonds are breaking, but it's not a true mechanism for the reaction. Okay, so we're going to get a new bond between the nitrogen of the amine and the carbon of the carbonyl. Okay, we're going to lose OH from the carboxylic acid and H from the amine to form H2O. And so H2O will be a byproduct of this reaction. All right, so what's our new product? So nothing changed here. So I'm just going to write R and my carbonyl. But this bond did break. The new bond is to the nitrogen of the amine. So my new bond, my amide bond to the amine, or to the nitrogen I should say, and we were generic, so R2, and byproduct is H2O. Okay. And this is my amide bond. Okay. Now the reverse of this reaction, the splitting apart of that amide bond with water, reverting it back to the carboxylic acid and the amine. Remember that splitting a bond apart with water is known as hydrolysis. So I'm going to label that. And just like the hydrolysis of the esters will be critical in the digestion of triglycerides, and the hydrolysis of the glycosidic bonds will be critical in the digestion of carbohydrates, the hydrolysis of the peptide bond, the amide bond, will be critical in the digestion of proteins. Okay, so a good reaction to note here. All right, so let's take a look at this reaction between amino acids then. So let's just pick some simple amino acids because I don't want to draw any fancy R groups. Whoops, my alpha carbon there, don't forget that. There we go. Let's make this one alanine. So the R group of alanine is a CH3. So I'll label that amino acid alanine, ALA. And let's have it reacting with, again, something simple. Let's make it glycine. Glycine's a simple one. So GLY to label that as glycine. All right, and again, I'm going to use some arrows. Remember, this is not a true mechanism, but it will help you, or at least I hope it will help you, to remember where that new bond is forming. Okay, so lone pair of the nitrogen, forming a new bond here. We're going to lose OH and one of the H's from the amine as H2O. Okay, so what does our product look like? We're going to have a free amine still from the alanine, our alpha carbon of alanine, and our carbonyl from the alanine. None of this part of the molecule changed. That's all going to stay the same. Okay, none of those bonds got broken apart. New bond starts here. Now the carbonyl of alanine is linked to what was the amine of glycine. So an NH. And then the rest of glycine is unchanged. Okay, so we've got our alpha carbon and our carboxylic acid. And don't forget, your byproduct is H2O. Okay, so this bond is more specifically referred to as a peptide bond when it occurs between two amino acids. So I'm going to write that here, and from now on you'll hear me refer to this bond as a peptide bond. Okay, so this bond here links those two amino acids together. And then to name this new molecule, we're just going to use the three-unit abbreviation for alanine and the three-unit abbreviation for glycine. That's the simplest way to do it, but you might also hear it called alanyl glycine. Okay. And when just two amino acids, let's write this down here, if it's just two amino acids that are linked together, we would say that the result is a di for two, a dipeptide. Okay, so this is the dipeptide known as alanyl glycine. When there's multiple amino acids linked together, you'll hear it called a polypeptide, or maybe even just a peptide. So I'll write both, polypeptide or just a peptide. It's not until we get to a large number of amino acids being linked together that we actually call a molecule a protein. It takes 50 or more amino acids to make it a protein. So let's write here 50 plus amino acids make it a protein. Okay, so you can imagine that this free carboxylic acid of this dipeptide, it's available now to react with the amine of another amino acid. Then you'd have three linked together. And then the free carboxylic acid of that amino acid would react with the amine of another amino acid, and you'd have four linked together, and on and on and on until you get 50 or 100 or hundreds of amino acids making up that larger protein. Okay, before we go on, very important to note, I'll do it in the blue pen here, that the side of your peptide or your protein that has the free amine, the free NH2, it's always going to be written on the left side. We traditionally write the structure of these peptides and proteins from the N, free nitrogen, to the C, free carboxylic acid end, and it's called the N terminus. The side of the peptide or the protein with the free amine, that is the N terminus, always written on the left. And the side of the peptide or the protein that has the free carboxylic acid is your C terminus, and that is traditionally going to be written on the right. Okay, so these are always written N to C. That's how we write the structure of our proteins, our peptides and our proteins. All right, let's go ahead and erase here. So the order of the amino acids is critical to the function of the protein. So let's just look at the dipeptide that we drew up on the board in the last segment of the tutorial, alanyl glycine. And let's go ahead and take that dipeptide and just switch the order of the amino acids so that we now have the dipeptide glycosylalanine. Okay, so then we're going to have on the N-terminus side, we're going to have a glycine, not an alanine. And on the C-terminus side, notice it now, we're going to have an alanine rather than the glycine. Okay, so this would be glycosylalanine instead of alanyl glycine. And these two dipeptides would not be the same. They would be different. Okay, again, now we have a different amino acid on the N-terminus side and a different amino acid on the C-terminus side. The order of the amino acids in a protein is critical to the function of that protein. You might remember at the beginning of this tutorial, we said that it is the structure of the protein that dictates its function. And just one amino acid being out of place is enough to cause a protein to lose its function. It may no longer be able to do its job in the body. And the order of the amino acids in a protein is referred to as its primary structure. I'm going to write that up here on the board. The primary structure. That refers to the order of amino acids in a protein chain. So again, if just one amino acid is out of place, the entire protein could lose its function. This is exactly what happens in the genetic disease known as sickle cell anemia. In sickle cell anemia, there's a 146 amino acid protein that has just one amino acid out of place. In place of what would normally be a glutamate with a hydrophilic, water-loving side group, is instead a valine, an amino acid with a nonpolar hydrophobic, that is water-fearing, side group. And it's this one change in that primary structure. Out of 146 amino acids, just one amino acid being changed results in a change to the overall hemoglobin protein. And that in turn results in a structural change to the overall structure of the hemoglobin carrying red blood cells. It causes them to sickle, as the name of the disease implies, sickle cell anemia. And these sickled red blood cells are less able to carry oxygen through the bloodstream. And that, of course, results in all of the effects of the disease, sickle cell anemia, including the anemia, because these sickled red blood cells have a shorter lifespan and die off more readily. So again, primary sequence, that primary structure, the order of the amino acids, very critical to the function of the protein. All right, we're going to go on, we're going to erase here and go on to talking about some additional levels of protein structure, the secondary, tertiary, and quaternary structures. So proteins are not just long chains of amino acids that flop around randomly. They have a much more specific structure than that. The next level of structure that we're going to talk about is the secondary structure. The secondary structure refers to the regular and repeating spatial organization of neighboring segments of the protein chain. It's already written on the board as well as on slide 4 for you. And these regular repeating patterns of the secondary structure are stabilized by hydrogen bonding. And that hydrogen bonding occurs between carbonyl units and NHs along the protein backbone. So what does that mean? Along the protein backbone. Okay, so let's write that. Let's write that this is stabilized by hydrogen bonding. I'll try H bonding between the carbonyls and NHs of amino acid backbones. All right, backbone chains. Okay, so what does that mean? So the backbone of any protein chain will be carbonyl, alpha carbon, nitrogen. Carbonyl, then the alpha carbon, and then the nitrogen. Carbonyl, alpha carbon, nitrogen. Okay, that's the repeating backbone chain. Carbonyl, carbon, nitrogen. Carbonyl, carbon, nitrogen. Carbonyl, carbon, nitrogen. Let's do a few more. Carbonyl, carbon, nitrogen. Carbonyl, carbon, nitrogen. Okay, and then I'll just write that this is continuing, because this is just a little backbone snippet from some longer protein that I'm trying to represent here. Okay, so what do we have? We have carbonyl, carbon, nitrogen. That represents one amino acid. All right, amino acid one amino acid in this protein chain. Carbonyl, carbon, nitrogen, that came from another amino acid, so I'll write amino acid 2. Then we've got carbonyl, carbon, nitrogen from a third amino acid. Carbonyl, carbon, nitrogen from a fourth amino acid. And carbonyl, carbon, nitrogen from a fifth amino acid. And then I just didn't want to draw any more, so I drew 5. That was enough. But we're talking about a protein, so we're talking about 50 or more amino acids in this overall chain. I just drew the backbone for 5 amino acids in the chain. So that's what I mean by the backbone. Okay, the carbonyl, carbon, nitrogen, carbonyl, carbon, nitrogen, that repeating unit from the amino acids. All right, so hydrogen bonding, how does it occur between amino acids, carbonyls of amino acids, and NHs of amino acids in the backbone chain? Well, we can imagine this carbonyl here would want a hydrogen bond to an NH. Okay, the hydrogen here maybe between amino acid 1 and amino acid 5. So let's indicate, and I'll just do it this way, that some hydrogen bond could form there, placing that hydrogen between the two highly electronegative atoms, nitrogen, the one that it's bound to, and attracted to the oxygen, okay, creating a hydrogen bond. Now that would cause this protein chain to bend, to fold in a specific way. And that is the secondary structure. Okay. Now the secondary structure really can be subcategorized into 2 categories, the alpha helix and the beta sheet. Okay, so with the alpha helix, you're going to get, let's go back to the black pen, you're going to see the hydrogen bonding occurring between the carbonyl of an amino acid and the NH of an amino acid 4 units down the chain. Actually what I drew here. Okay, so the hydrogen bonding occurs between the carbonyl of one amino acid and the NH of an amino acid. 4 units down the chain. And this results in a spiraled configuration, like a slinky. So it's right here. Results in a spiral, like a slinky. And that spiral will have a right-hand turn and all of the R groups, all of the side groups, will be on the outside of the spiral, the coil. R groups on the outside of the spiral or the coil. Okay, so we're running out of room here. Let's see how I can do fitting this in over here. Okay, so it's really what I've drawn up there, but I'm going to bring those 2 amino acids together and show the coiling, the spiraling. Okay, so let's say this is amino acid 1, 2, 3, okay, and I said a right-handed turn, so imagine, take your right hand, not your left hand, take your right hand and point your thumb in the direction of the protein chain. So I'm going to do N to C, so N is my first, and C will be down here. I'll go down to 10. Okay, your fingers are going to show you the direction of the spiral or the coil. Okay, so I'm trying to show that my amino acid chain, my protein chain, is coiling out and around like this. Okay, I hope that helps. Okay, so then 4 would be coming out, 5, and then 6 would be going into the plane of the whiteboard. And then we've got 7, I'll go all the way to 10, 8, 9, 10. Okay, so we're doing N terminus up here, all the way down to our C terminus down here off of amino acid number 10. Okay, so let's do our hydrogen bonding in this pink just to make it really stand out. So that means that we're going to have hydrogen bonding occurring between amino acid 1 and the amino acid 4 units down the chain. So 1, 2, 3, 4. So amino acid 1 and amino acid 5. Okay. Amino acid 2 would be hydrogen bonding to 1, 2, 3, 4. Amino acid number 6. Okay. 3 to 7. And let's see, 4 to 8. I knew I wasn't drawing that right. Let's see. Eek. Getting got in the way there. 4 to 8. 4 units down the chain. Okay. And then 5 to 1, 2, 3, 4 to 9. See how they're in a nice position for the hydrogen bonding to form, stabilizing that spiraled structure, that alpha helix. Okay, 6 to 1, 2, 3, 4 to 10. Okay, so I'm kind of getting messy here, and so on down the chain. Okay. All right, and then the R groups would be sticking out this way. Okay, so if you were looking down that coil, if your eye was up here and you were looking down that coil, let's draw it here, your R groups would be sticking out like so. Whew, these things are hard to draw. Hopefully my drawing makes sense to you, and the definition as well. Now we don't have room, so I'm going to erase here, but we also want to talk about the second subcategory of the secondary structure, which is the beta sheet. So let's erase here and go on to beta sheet. Okay, in the beta sheet, which again is a subcategory of the secondary structure, we have hydrogen bonding occurring between the carbonyls and the NHs of amino acids that are in neighboring segments running anti-parallel to one another. So let's write that up here. So we have hydrogen bonding, H bonding, occurs between carbonyls and NHs from neighboring segments of the protein chain that are running anti-parallel to one another. Okay, so what does that mean? So again, we're going to do that backbone, that carbonyl, carbon, nitrogen, carbonyl, carbon, nitrogen. And let's start it here. Okay, carbonyl, and this is just continuing out this way. Carbon, nitrogen. Carbonyl, carbon, nitrogen. Carbonyl, carbon, nitrogen. Okay, remember that this is the repetitious backbone of the protein. That's the backbone from all those different amino acids being linked together. Let's do one more. Carbonyl, carbon, nitrogen. Okay. And then let's say that protein chain, continues to do some things. Maybe it goes out here and does some alpha helixes and some different things, and then it loops back around. Okay, now it's going to be running anti-parallel to the first segment. Okay, so anti-parallel. So we're going to have, let's see if I can do this nice and neat. It's not easy to do. Do it a little further down. There we go. We're going to have, go up, carbonyl, carbon, nitrogen. Carbonyl, carbon, nitrogen. Getting kind of messy here. Carbonyl, carbon, nitrogen. Okay. So hopefully you see why that is considered to be anti-parallel. So we were running carbonyl, carbon, nitrogen this way. Okay. Now we're running carbonyl, carbon, nitrogen this way. So anti-parallel segments. So it's the same protein chain. It went out here and it did a bunch of other things, came back around, and now is running anti-parallel to the first segment that we drew. Okay, so continue with that. Carbonyl, carbon, nitrogen. And then we'll just indicate that it's continuing because, again, this is a protein chain. This has a lot of amino acids in it. We're not going to draw them all. We're just going to draw little snippets so that we can indicate what the beta sheet means. All right, so hopefully you can already see all this opportunity for hydrogen bonding that would stabilize this beta sheet structure. Okay, you can see it here between the carbonyl of this amino acid and the NH of this amino acid. You can see it here, all in perfect position to hydrogen bond, and it's all that hydrogen bonding that stabilizes this regular repeating pattern known as the beta sheet. All right. Okay, so let's go ahead and erase here, and we're going to go on to talk about the next level of protein structure. It's not this simple. It's not just primary and secondary structure. There is a tertiary structure and a quaternary structure as well. The tertiary structure of a protein refers to the way that that protein will bend and fold upon itself to make up its final three-dimensional shape. And the tertiary structure is stabilized by interactions that involve the R groups, that is, the side chains of the individual amino acids. So remember, in the secondary structure, the stabilizing interactions were just hydrogen bonds that occurred between carbonyls and NHs of backbone amino acid chains. Now we're talking about interactions that will involve the R groups, the side chains of the amino acids. That was not involved in the stabilization of the secondary structure. So let's write up here that the tertiary structure is going to be stabilized by interactions involving the R groups, that is the side chains of the amino acids. Okay, so that means that those interactions can be quite varied. Let's talk about those different interactions that can occur to stabilize the tertiary structure now. Well, hydrogen bonding. We already talked about it with the secondary structure. Hydrogen bonding can be involved in stabilizing the tertiary structure as well. So it's right here, H bonding. But in addition to that, there can also be hydrophobic interactions, where a group of hydrophobic side chains coagulate together and create a hydrophobic pocket that sticks together and stabilizes the structure of the overall protein. So it's right here, hydrophobic interactions In addition to that, there can be salt bridges, which occur between the positively charged basic side groups and the negatively charged acidic side groups. Those positive and negative charges form a salt bridge, like an ionic interaction. Okay, so those are called salt bridges. And last but not least, also disulfide bonds. Now, a disulfide bond is actually a covalent bond. And a disulfide bond can only occur between two side groups of cysteine units placed close together. So let's write here disulfide bonds. Okay, and I think that this is best shown with a picture. So I'm going to do my best to draw a picture of a protein chain. Okay, and the tertiary structure will involve the secondary structure. So it's going to have some beta pleats and some alpha helixes, one or the other or both, throughout. So I'm going to indicate those in my drawing. I'm going to start out with a beta sheet over here, and then we'll have a loop coming up here, and we'll come back around to finish our beta sheet, and then maybe we have a loop here, and then maybe we have some alpha helixes, maybe a couple of them. And then let's make a final loop over here, and then just indicate that's just a portion of our protein chain. And let's show these interactions in a different color. Let's pick the pink pen because I just really want things to stand out. So we already know that we're going to have hydrogen bonding, holding the beta sheet and the alpha helixes together. So let me just indicate some dots for my hydrogen bonding there. And we talked about those earlier in this tutorial, so I'm not going to talk about those again. and kind of bump that. Okay. All right, so let's show first, let's show our hydrogen bonding. And remember that in the tertiary structure, this is occurring between side groups. This is going to involve side groups. So let's say that we have a hydrogen bond right here. And let's say it's between a serine unit, because we know that has an OH. And let's just make it between the carbonyl of a backbone amino acid. So the serine group, the CH2OH, can hydrogen bond to a carbonyl of a backbone amino acid. So here's our R group getting involved here with this hydrogen bonding. So I'll label this hydrogen bonding between the serine side group and a carbonyl of the backbone. Okay, so that would stabilize this big fold that we have here of the protein coming around, looping around, folding back around so this beta sheet can line up over here. So a hydrogen bond stabilizing that portion of the tertiary structure. All right, next let's do hydrophobic interactions. Let's go to the blue pin just to hopefully make it stand out a little bit from what we did over here in pink. So over here maybe we have a hydrophobic pocket where we have a bunch of hydrophobic non-polar side groups from different amino acids coming together, coagulating together, sticking together because they don't want to be oriented out toward the aqueous environment. So let's say that we have, let's see, valine is a non-polar side group. Let's put a couple of those here just because that's an easy one to draw. And let's put an alanine, that's a nice easy one. We know the side group of alanine is a CH3, again, a non-polar side group. And let's put another alanine here. Okay, so then I'll label this hydrophobic interactions between two alanines and two valine R groups. They're coagulating together there because they are all nonpolar hydrophobic side groups. Stabilizing again this bending and folding here in the protein structure. Okay. All right. Next, let's do a salt bridge. Okay, so I'll switch colors. Let's do green. Just trying to make these stand out a little better. Okay, so let's say right here that we have a CH2CO minus. So you have an aspartate side group. The side group of aspartate is CH2COOH, but remember that the acidic side groups are deprotonated at physiological pH, giving it a negative charge. And then over here, let's say that we have a lysine, which has a CH2, it has 4 of them. It's going to be kind of hard for me to draw, so I'm just going to write CH2, 4 of them. And then at the end of that, it's got an NH3+. Okay, it's got an amine group, but remember that the basic amine groups are going to be protonated. So it's going to be NH3+, it's going to be charged at physiological pH, and that positive charge and that negative charge attract in what's called a salt bridge. It's like an ionic interaction, but it's between amino acids that are stuck together in this overall protein chain. Okay, so they're within the same molecule. So this is a salt bridge. So let's label this. Salt bridge between aspartate and lysine R groups. Okay. And last but not least, let's do an example of a disulfide bond. So a disulfide bond has to occur between 2 cysteine residues. And cysteine has a chain of carbons with a thiol functional group on the end, which is an SH. And 2 thiols can react to form the disulfide bond. So let's say that we have a cysteine residue here and a cysteine residue here. So it's going to end up with an SH. SH. Okay. When they're in close proximity, these 2 residues can react to form the disulfide bond, which is a covalent bond between those 2 sulfurs. Okay, let's label that here. Disulfide bond between 2 cysteine R groups. So again, we're talking about an interaction involving the R groups that's going to stabilize this specific bend and fold in the protein chain. So it seems random. It seems like you're just taking a piece of paper and you're crumpling it up, but it's not. It is not random at all. This tertiary structure is very specific. And again, just like all the structures of the protein, the overall structure of the protein, this tertiary structure is very important to the function of the protein. With the loss of this tertiary structure, we would just have randomly, moving about spaghetti-like strands in this protein chain. So this tertiary structure is really very critical to the overall function of the molecule. As random as my bends and folds might look to you, they're very, very important to the overall function of the protein. All right, so let's go ahead and erase here. Some proteins are composed of 2 or more separate chains that come together to form the final functioning protein. This is known as a quaternary structure, and not all proteins have it. Some proteins are just composed of a single protein chain. Others are composed of multiple chains that again come together in this way to form the final functioning protein. The quaternary structure is stabilized by the same types of interactions that we saw in the tertiary structure. Okay, so let me write that here. Stabilized by the same interactions just discussed for the tertiary, all right, 3' structure. Okay, so hydrogen bonding, disulfide bonds, hydrophobic interactions, and salt bridges. All of those can also be utilized to stabilize the quaternary structure. So a great example of a protein that has the quaternary level of structure is hemoglobin. Hemoglobin is composed of 4 separate protein chains. 2 of them, so let's write here for example, EX for example, hemoglobin. It's composed of 2 146 amino acid long protein chains and 2 141 amino acid long protein chains. And then those 4 proteins have to come together to make up the final functioning hemoglobin protein molecule, protein structure. 4 proteins must come together, let's write that here, to make up the final functioning hemoglobin. Okay, so we'll indicate this just with kind of a blob here for one of my protein structures, and then a blob here for another one. and a blob here for a third, and another blob here for a fourth. And then for those interactions, you would see interactions occurring. I'll just use dot dot dots to indicate that. Not that they're all hydrogen bonds. They can be salt bridges, disulfide bonds, hydrophobic interactions, and they would be occurring between those four separate chains of the hemoglobin molecule to keep them together so they don't come apart, so that we have our final functioning hemoglobin structure. Just as the primary structure is critical to the function of a protein, so too is the secondary, tertiary, and quaternary structure. And if a protein loses its secondary, tertiary, and or quaternary structure, we say it has been denatured. And a denatured protein cannot perform its function. So what kinds of things can denature a protein? Anything that causes a disruption to those stabilizing interactions that we just talked about for the secondary, tertiary, and quaternary structure. So that would be anything that can disrupt hydrogen bonding, anything that can disrupt the salt bridges, the hydrophobic interactions, and the disulfide bonds. So I've listed a few things that can cause a protein to become denatured up here on the board, and these are also listed on our last slide, slide 5 of the notes. And we'll just talk about each briefly. So first, heat and ultraviolet radiation. These can cause denaturization by increasing the kinetic energy of the protein chain and thus ripping apart the weaker intermolecular forces of attraction that are stabilizing the secondary, tertiary, and quaternary structures. Mechanical agitation, such as beating an egg white, introduce air bubbles into the protein. And these air bubbles will disrupt hydrophobic pockets that are stabilizing the tertiary and quaternary structure, again causing the protein to denature. Detergents have both hydrophobic and hydrophilic portions. The hydrophobic portions of detergents can interrupt the hydrophobic pockets that stabilize the tertiary and quaternary structure of a protein, therefore causing denaturization. Polar organic solvents such as ethanol will disrupt hydrogen bonding, which is crucial to the secondary, tertiary, and quaternary structure of a protein, thus again causing denaturization. Changes in pH, so adding acid or base, can either protonate or deprotonate our acidic or basic side groups, thus disrupting our salt bridges. If one of those units is no longer charged, we're not going to have an attraction there between the positive and the negative. No longer exists. And finally, addition of salts can do the same. It can also disrupt salt bridges and denature our proteins. Okay, so just a quick summary there of denaturation. Last thing I want to say about it is that the primary structure is not susceptible to denaturization, because in order to lose the primary structure, that is the sequence of the amino acids held together by the peptide bonds, a chemical reaction known as hydrolysis must occur. Hydrolysis, remember, is a splitting apart of a bond by water. So in this case, the splitting apart of the peptide bond by water. The only way to lose a primary structure is through hydrolysis of those peptide bonds, a chemical reaction. Okay, so the primary structure is not susceptible to denaturization like the secondary, tertiary, and quaternary structures are. All right, we will conclude our tutorial 23 right here. I hope that you will consider tuning into problems at 23, where we will work lots and lots of problems to hopefully help you solidify all of this knowledge from proteins. Thank you very much.